We have had an interest, I've had a personal interest in the development of so-called oncolytic virotherapy for at least 20 years now. There is a group of viruses, or a range of different viruses, that have the very unusual biological characteristic of being able to grow very effectively in tumor cells in the laboratory, but not growing at all in normal cells, or hardly at all in normal cells. What we're now doing in the clinic is trying to exploit some of those therapies to see whether or not we can use them as cancer-specific infections that can kill tumor cells. And we're also now understanding, and have understood for some years, that they can also activate the immune system. The leading agent in this field is a drug called Talimogene laherparepvec, which is a replication-competent herpes simplex virus type 1 that has been genetically modified so that it is incapable of growing in normal cells, but is capable of killing tumor cells. And this virus has been genetically altered so that it expresses the human protein granulocyte macrophage colony stimulating factor. In a phase 3 clinical trial in melanoma, we demonstrated a positive outcome relative to the control arm, which was subcutaneous GM-CSF administered by the patient. And what we saw was a significant improvement in the durable response rate with an odds ratio of nearly 9 and a difference of about 17% versus about 2% in durable responses in those patients. What we also saw in a subset analysis in stage 3B, stage 3C and stage 4 M1A disease in melanoma was a significant improvement in overall survival in those patients. That led to the approval of this drug by the FDA and by EMA in 2015. This drug is now licensed. It is prescribed by doctors across the world for the treatment of melanoma. We are very interested in developing this drug in combination with other immuno-oncology drugs. So there are phase 3 trials running in melanoma and a phase 1B trial running in head and neck cancer that will transit, we hope, into a phase 3 trial in which TVEC is being combined with the anti-PD-1 immuno-oncology monoclonal antibody pembrolizumab. So I think for oncolytic virotherapy, we're at the start of a very exciting journey in which we will begin to develop these immunotherapeutic agents in combination with other IO drugs, again for patient benefit, initially in patients with relapsed disease, but hopefully ultimately in earlier stages of disease to increase cure rates.